Hello, and thank you for joining us. This is Scott with Eurofins, bringing you our exclusive on-demand webinar titled Making Sense of Presumptive Pathogens, Understanding Rapid Methods and Confirmations. I'd like to start by giving a brief introduction to our featured guest. Dr. Christopher Crow is the Senior Director of Science for Eurofins Microbiology Labs. In his role, he manages the R&D team as well as provides technical leadership and client and operational support across the entire network of Eurofins labs. Dr. Crow holds his PhD in immunology, a bachelor's degree in biochemistry, and has maintained over 15 years of experience in the diagnostics and food testing industry, with previous roles at Gold Standard Diagnostics, Hygiena DuPont, and Hardy Diagnostics. His presentation here will aim to cover a background review of how we have come to report results as presumptive, the current state of rapid and confirmation testing, and possible future direction. Along the way, he will clarify commonly confused testing terminology and build context and perspective around what these results mean to you. Dr. Crow first gave this presentation at an open house event at Eurofins Microbiology Laboratory in Tustin, California, where the talk garnered much attention from a varied group of industry professionals. I'm delighted that he is here to provide this once again for a broader audience. All right, with that, Chris, I'll hand this over to you to begin your talk.
All right, thanks, Scott, and thank you for the kind introduction. And thank you to everyone joining us and for your interest in the webinar. As Scott said, today's topic is to try to understand the term presumptive positives, where that term came from and why we use it. And along those lines, to really elucidate the role for using rapid methods and cultural methods in pathogen testing within the food industry. So as part of that topic, today's objectives are laid out here. So first, we'll discuss a little bit about where the term presumptive positive came from and why people started using it in the first place, and then also why we continue to use it today. We'll also touch on why we always do cultural confirmations when we get a presumptive positive. And then we'll switch gears a little bit to talk about what it means when you have a presumptive positive and you go on to cultural confirmation testing, but that presumptive positive does not confirm by cultural methods. And more importantly, what you should do or how we should be reacting to these non-confirming presumptives. So it makes sense to kind of start with a little bit of a history of where the term presumptive came from within food pathogen testing. And so today, when we think about food pathogen testing requirements, most of us would say that the regulatory agencies responsible for those requirements are either the USDA or the FDA. But interestingly, the first pathogen monitoring requirements came from NASA and not the FDA or USDA. And they set requirements not only for pathogen monitoring, but also for toxins and even crumbs. And to me, crumbs seems like a kind of a silly thing to have testing requirements for. But I guess when you think about NASA and the space shuttles and all the expensive equipment on a space shuttle, you probably don't want crumbs floating around in zero gravity that could get into the equipment and mess it up. So I guess in that sense, it makes a good deal of sense that they would be regulating crumbs as they were regulating pathogens. Irregardless, I think the first requirements for pathogen testing came from NASA, and that was all the way back in 1962. And then for roughly 30 years, all pathogen testing was done using cultural methods. We didn't have rapid methods available to us. And for those who are familiar with the cultural methods that are available, you know that it's kind of a long and arduous process. And so this schematic timeline here on this slide is really just meant as an example. And depending on which pathogens you were testing for, it could be a couple days longer or shorter. But I think the point remains that you generally took 3-4 days to get to a point where you would have a Petri plate that either would or would not have your pathogen on it. And if there was no growth on that plate, great, now you're 3 days in and now you can release it as a negative result. But if there was growth, Then you go on to do biochemical testing in order to identify whether that growth was a pathogen or some other organism. And so that could add as much as a whole nother week on top of that timeline. And so for those that are in industries like fresh produce or fresh seafood or meat and poultry, you probably recognize that there aren't necessarily 2 weeks of shelf life available to be doing pathogen testing. And so really that limited how broadly pathogen testing requirements could be applied. And in the early 90s, we actually saw the first commercialization of rapid pathogen test methods. And for the sake of this talk, I broke it down into really two categories. And first is immunoassays. And immunoassays are tests that are mediated by antibodies. And so the antibody will bind to the pathogen if the pathogen is present, and the antibody will then provide a signal that can be detected. So if there's a signal, you know you have pathogen. If there is no signal, no pathogen. Uh, but immunoassays have always been limited by antibody specificity. So unfortunately, a lot of antibodies tend to cross-react. So not only with the target of interest, but other closely related bacteria or organisms. And so even immunoassays that are on the market today, we see, let's for instance use salmonella as an example. An immunoassay for salmonella may also cross-react with species such as citrobacter, which is not a pathogen, but will nonetheless give you a positive result. And so for that reason, immunoassays are somewhat limited. And for the rest of this talk, we're not going to spend a lot of time on immunoassays. Instead, we're going to focus on the second bucket, which would be molecular methods. And the prototypical molecular method is PCR, polymerase chain reaction. And so molecular methods work by amplifying very specific targets within the organism's genome. So it's a genetic DNA-based test. And as a result, it has a lot more specificity than immunoassays. So you have a much lower chance of getting a cross-reactor or something like that. Now, when PCR was initially discovered and commercialized, the assays were what we call gel-based. And what I mean by that is we could amplify the DNA targets that we were looking for, but we had no way to detect them within the assay itself. And so once the assay was completed, you would take that assay and you would load it onto an agarose gel, and then you would interpret that gel by eye to determine whether the pathogen was present or not. So there was a fair amount of human subjectivity that went into the results. So with these new test methods coming onto the market in 1992, the organization that is today known as IAFP brought together a group of experts from all walks of industry to kind of understand how these rapid methods could be used within the paradigm of food pathogen testing. And really you had a bunch of competing viewpoints. So on one hand, you had the test kit manufacturers. And from their point of view, these rapid methods, in addition to being faster than culture, were more sensitive, more specific. And, from their point of view, these new test methods should be considered the gold standard for pathogen testing. On the other side, you had the food industry who heard that these test methods were more sensitive and had a lot of concerns about that. And I would even say maybe beyond concerns, maybe a little bit of mistrust in the technologies because, a more sensitive test presumably then leads to more positives. And more positives are going to lead to more scrap or rework, increased costs, potentially more recalls and lawsuits and things like that. And in addition to that, you also had this concept, particularly with PCR, where there was this concern that because the assay was only looking for DNA, that you would be detecting dead cells as well as live cells. So if you had a product that had salmonella in it, but it went through some intervention or a kill step or something like that, it would be safe for consumption. But because the DNA was still present, the fear was that PCR was still going to be detecting it, and you're going to be considering it to be pathogen positive. And so there were a lot of concerns from industry about how to apply these newer methods. And the agreement that's had kind of been reached was that if you get a negative result, then everybody's happy and we're all good to go. But in the case of a positive result from one of these rapid methods, the thought was it should be reported as presumptive until you confirmed it using a validated culture method. And so in the US, most of the cultural methods that we use for confirmation come from either the FDA's BAM or a USDA MLG or the compendium. There's other sources, but I would say those three are probably the bulk of our cultural methods. Globally, ISO methods play a larger role as the validated culture method. But regardless, the point would be that the positive results from a pathogen test would not be assumed to be true positives until they were confirmed using a cultural method. And so what are the implications of structuring your test method that way? Well, first of all, you get the main benefit of having that rapid method, because now instead of waiting several days, you can get a result in 24 to 48 hours. And in fact, nowadays, it's even, depending on the test and the organism, it's even less than 24 hours before you can get a result. And if that result is not detected, then everybody's happy and we can release the product, much sooner than we could have if we were using cultural methods. On the other hand, if the rapid method returns a positive result and pathogen is detected, as a producer, you guys have a choice. First of all, you could choose to trust that rapid method result. So you could choose to act on the PCR result without going through cultural confirmation. But you can't overturn that result if you have a reason to doubt it, or even if you don't have a reason, if you just hope that it's wrong, you can't overturn that result unless you go through the cultural confirmation method, which then adds several days to testing. And so, one thing you don't want to do is if you get that detected result with your PCR reaction, you can't just repeat the test until you get an answer that you like. That sort of practice of testing into compliance generally doesn't go well. So the requirement is if you get a pathogen presumptive result from your rapid method, the proper thing to do is to go on to cultural confirmation if you aren't going to treat that positive as a positive and act on it. So we've essentially set up a dichotomy here where we trust the negative results from a rapid screening method, but we confirm a positive result with a cultural method. And so now we've had PCR and rapid methods for about 30 years. And in addition to all the experience that we've gained using these platforms, the platforms themselves have developed. So the PCR assays that we have today are not only faster and they're more automated, but they're also much more sensitive and specific than the original gel-based PCR analyses that were available, 30 years ago. And so over time, gel-based PCR analyses gave way to melt curve PCR, and melt curve gave way to real time, and now we even have isothermal methods. And I would argue that right now, the state of the art is that real-time PCR and isothermal methods are the gold standard and used most broadly across food testing. There still is a limited role, I think, in some situations in some food matrices for melt curve PCR. I would argue that gel-based PCR, what we know now about sensitivity and specificity, it's probably inappropriate for labs to be using gel-based PCR. So if you're testing with a lab that uses gel-based PCR, I would say they're probably borderline malpractice at this point to continue using those platforms with such better technology available to us. And while I'm not going to go through the chemistry of each of these iterations and each of these assay types, the one point I will make is that by going through this evolution, we've essentially removed the human subjectivity because we no longer have to run the results out on a gel to be interpreted by a technician. So nowadays, the plus or minus calls, the positive or negative calls, are made by software algorithms. So the reactions generate a fluorescent signal, and the computer collects that signal, and a software algorithm, does statistics and linear regression or whatever it does to generate a positive call or a negative call. So that's kind of a double-edged sword. It's good in that it removes human subjectivity from the equation. It's bad in that software algorithms can be wrong too, and they're not perfect. And I want to go through just a very brief example of why that is. So this is a typical PCR, real-time PCR graph. And for those that are not familiar with interpreting this data, you'll note very quickly there are two lines on the graph. This bottom line, the black solid line, is an internal positive control. So nowadays, all PCR assays contain an internal positive control. And what that does is it allows the operator to know that the PCR reaction worked. And what we're looking for in our positives is this kind of sigmoidal or S-shaped increase in signal. So that shape to that curve tells us that there was amplification of the target gene. So in this case, the internal positive control amplified. And then the kind of red dashed line here is your pathogen target. And so again, in this example, you see this sigmoidal amplification, which indicates that that pathogen also amplified. So this would be a positive pathogen result. And if we contrast that to a negative result, you'll notice here again, your internal positive control still has the amplification, but your pathogen target is more of a straight line throughout the run. And that would be considered a negative because you don't have this sigmoidal amplification that you see in a positive. So I think it's pretty obvious by eye the difference between a positive and a negative. And as a result, the software algorithm in 99% of the cases is able to distinguish between a positive and negative. But I want to share a curve here, a case where that's not necessarily true. So in this example, again, your internal positive control amplifies and looks good. And I would argue that this pathogen target looks pretty flat to me. So I would expect this to be negative. But you'll notice here at the beginning, there's a little bit of I guess we'll call that a glitch here at the beginning. And if you look right about here, you can kind of squint and see maybe a little bit of an inflection point. And those two features, I think, combined, tricked the algorithm, or maybe not tricked the algorithm, maybe we'll say triggered the algorithm into calling this a positive curve. And so what does that mean? Well, we don't want labs to be overruling the algorithm, right? That introduces reintroduces the human subjectivity, and it becomes a slippery slope as to which curves you trust and which curves you don't trust. So when the algorithm says this is positive, it has to be treated as a positive and sent on to cultural confirmation. Now, in this case, this particular sample did not confirm, it was negative. But the problem there is now that you're introducing this variable, you're delaying getting the result to the client. And so obviously, if you have product that's waiting to go out the door and the algorithm returns this sort of result, now you have an unnecessary delay as you wait for cultural results to come back. But I think I chose this as an example to say that even despite 30 years of development, PCR is still not perfect. There's still occasionally issues with it. And so that's why we continue, still today to have to go on to cultural confirmation. And so how do we proceed, or how do we think about this? And for me, I return to the analogy that everybody's probably familiar with, and that is that pathogen testing is really like searching for a needle in a haystack. And if you'll allow me the latitude here, I'm going to take that analogy and I'm going to stretch it to its limits. Actually, I might even stretch it a little bit beyond its limits. But for me, if we use this analogy, I would think of rapid methods like PCR similar to an X-ray or a metal detector. And so as I said on the previous slide, over the last 30 years, we've made our metal detector faster, we've made it more sensitive and more specific, but as we saw with the algorithm, it's still not perfect, and it probably is never going to be perfect. But I'd contrast that with cultural methods. So for me, in this analogy, cultural methods would be like reaching into that pile of hay and trying to find the actual needles. And so when you think of it that way, you can imagine your likelihood of actually finding the needle is going to depend on a lot of factors. It's going to depend on how big is the haystack? How many needles are in the haystack? Are those needles evenly distributed throughout the haystack? And what I hope you're probably coming to realize here is that you can have situations where these two methods are going to disagree. And so really there's three possible scenarios here with this analogy. So first you have your rapid method PCR or a metal detector, and you wave your metal detector over the haystack and it doesn't beep. Your rapid test is negative. So as we said before, we trust the negative results from our rapid tests. We don't do cultural confirmation. We report that out as not detected, and I think everybody's happy with that result. In another scenario, we take our metal detector and we wave it over another haystack, and the metal detector starts beeping, right? So now we presumptively have needles in this haystack, right? So the PCR result has given us a presumptive positive. So what do? We move on to cultural confirmation, or we start going through the haystack to try and find these needles. And as luck has it, we reach in on our first handful, we pull out a couple needles, and so that confirms what we had with the metal detector, and we reported out that we've detected our needles in our haystack, or we've detected salmonella in our product, or whatever the pathogen might be. And again, well, nobody's happy with that, but everybody understands that what's going on. But you can also envision this third scenario. So you take your metal detector, you wave it over the haystack, and it starts beeping. So presumably you have needles in there. So it's a presumptive positive again. But now when you start reaching into that haystack, for whatever reason, after digging through a bunch of handfuls of hay, you're not finding any needles. And so what do we do in a situation like that? And so from a pure regulatory standpoint, the regulatory agencies say that if the cultural confirmation is negative, then it can be reported as not detected. So if that's all we're focused on, then this result is reported as not detected, and we say, well, this PCR result must have been wrong. But what I'm hoping this analogy helps illustrate is that there's probably scenarios, there are definitely scenarios within pathogen testing, where that presumptive result was correct, and we just missed it with the cultural confirmation. And so a non-confirming presumptive result does not necessarily mean that your screening method, your rapid test, was giving you a false positive. And I think that's a very important take-home point here. So the next question then that follows is, why would you get a different result between your rapid method and your cultural method? And because they're giving different results, which one do you trust? And I'm saying trust, meaning going beyond just the regulatory question. Which one do we actually believe is the correct answer? And as I said, no rapid method's ever going to be perfect, but I think it's also important to recognize that cultural methods are not perfect either and can yield incorrect negative results. And so I pulled a graphic. This is from a different presentation, but I think it's applicable here, of why a cultural result, that is where you fail to detect the pathogen, might be incorrect and be giving you a false negative. So first, you could have an overgrowth of non-target organisms that are crowding out or obscuring your pathogen. Second, you can have a pathogen where it has an atypical phenotype. And the example I always think of here is Listeria. So when we look at some Listeria confirmation plates, Listeria is expected to cause a blackening in the media. So it produces kind of a blackish, grayish colony. And one of the Eurofins labs not too long ago had an isolate of Listeria that did not blacken the media. And so we were fortunate in that that was the only colony on the plate. And so when we tested it, it confirmed as Listeria. But you can imagine if these first 2 points are combined and you have an atypical pathogen and it's surrounded by other non-target organisms that you can't tell the difference between the two, you're going to miss that pathogen on that plate. Another possibility is that the organism just isn't going to grow in vitro. And so there's this whole concept of viable but not culturable, VBNCs. And that topic could be an hour-long webinar in itself. But the bottom line is that for whatever reason, some reasons are fairly well understood, some reasons not so well understood, the organism just does not grow well in vitro. So it's not dead. It's very much alive. It's probably still capable of causing disease, but for whatever reason, it is not able to grow on a Petri plate. And there's many reasons again why that could be the case. It could be something as simple as it's not as resistant to the antibiotics in the plate as a typical strain would be, and so it just isn't able to grow. And then a final reason would be that the organism is below the limit of detection for cultural methods. And so we talked about how rapid methods are more sensitive than cultural methods. So you can imagine a scenario where the organism has grown up to the point where it's at the limit of detection for the PCR method, but it's still below the limit of detection for culture, and therefore it gets missed. And that one I would argue is probably a little less likely, but certainly there are reasons why the cultural result might be wrong. And so which result do you trust? You have now 2 methods. Neither of those methods are perfect, and they're giving you different answers. And you have to choose one to make a decision on, right? And if you were joining this webinar in hopes that I was going to give an answer to that question that would be definitive and make your life easier, then I'm sorry to disappoint you. But there is, in my opinion, not a one-size-fits-all answer to that question. And so what I would encourage you to do is to talk to your lab about these things, because the lab may be able to give you further information than what's reported out on a C of A. What did we see during culture confirmation? What did the PCR curve look like? Information that may help you make a decision as to which answer you want to trust. And so to kind of drive home this idea that there is no one-size-fits-all answer, I thought I'd throw in a hypothetical scenario here. So let's imagine that you have submitted 10 lots of product to your testing lab, let's say over the course of a month or the course of 3 months. And this subset of 10 lots of product all came back with a presumptive PCR result and a negative cultural confirmation. Now, as I indicated earlier, from a regulatory standpoint, you would be 100% justified to trust those negative culture results and release all 10 lots of that product. But let's say hypothetically that I have a crystal ball, and in my crystal ball, I learned that one of those PCR results was correct and the cultural result was a false negative. So we're talking 1 out of 10, 10% of my non-confirming presumptives were actually true positives. Now, my crystal ball is not very good, so it's not going to tell me which lot that is. Are you still okay, or are you still comfortable releasing all 10, knowing that it's going to prevent you from having to scrap or rework 9 otherwise good lots just to prevent one lot that might be contaminated with a pathogen? Or conversely, are you willing now to scrap 10 lots just to make sure that that one bad lot does not go out into commerce? And does your answer change if instead of 10%, it's 3 out of 10? Or what if it's 5 out of 10? Or what if it's 8 out of 10? And I think that if you put a bunch of smart people in a room, they'd all have a different opinion as far as what percentage PCR results are probably right versus cultural results. But I think that most people would probably agree that it's not cut and dry 100% one way or the other. And unfortunately, I don't have a crystal ball, and nobody has a crystal ball. But I think this is where having that conversation with the lab, understanding what's going on in that sample, what they saw in cultural confirmation, what they saw in PCR, can help you to decide which results you want to trust. And at least with Eurofins, I can say that not only do we have expert microbiologists in all of our labs across the country, but we also have a national technical team that can weigh in on this. In fact, we exist to weigh in on examples like this, and so we can try to help digest what's going on in these samples. And so I want to spend the next few minutes talking through a case study where we did just that with one of our clients. So this client was a large multi-site food manufacturer, and we switched all of their plants over to a new method. So they were doing Listeria species environmental testing, and they had been using a melt curve based PCR, which is an older technology. And we switched them over to our own proprietary back gene real-time PCR method, which is much more sensitive than that melt curve assay. So what happened was they started getting a lot more presumptives, which you would expect having a more sensitive assay. But they also noted that a lot of those additional presumptives weren't confirming. And so you can imagine that was becoming quite disruptive at the plants. So they asked us to investigate. So what we did was we took one plant, and over several months, we took all samples that were received by that plant, and we tested them not only by our real-time PCR method, but also by their previous method. And then any presumptive results from either method were sent on for cultural confirmation. Now, in parallel to us doing that on the testing side, the client was acting on all presumptive results. So because these were environmental samples, they didn't want to wait around for cultural confirmation. So they were doing whatever sanitizing their SOPs required based purely on those presumptive results. So what did we see in this study? So first of all, you'll notice that in the first month, they submitted 113 samples, of which our real-time PCR method picked up 16 presumptive positives. You'll notice that their previous method only picked up 4 presumptive positives. So you can see right away there's a difference in sensitivity between these two methods, but you'll note that only 5 presumptive positives confirmed. So a couple points here. First of all, that means that there was actually a confirmed positive result that their previous method had missed. But also it means that only 30% of the presumptives by real-time PCR were confirmed. So basically 2/3 of these samples failed to confirm, which you can understand why the client would be frustrated by that. So we go on into the next month, we get 17 presumptive positives. Their previous method only gets 11, and only 13 confirmed. So again, same trend. Their previous method actually missed 2 confirmed positives, but we've improved our confirmation rate a little bit. We're up to 76%, right? So that's a pretty good improvement over 31%. But if we broke that data down even further and looked week by week, you would actually note that all 4 of the non-confirming presumptives occurred in the first or maybe the second week of November. And so from the middle of November on throughout the rest of the study, you'll notice the real-time PCR confirmation rate was 100%. So in about 6 weeks, the confirmation rate went from 30% to 100%. And that lasted throughout the study. And you'll also notice that even though the confirmation rate improved, if you look at this last month, their previous method continued to miss confirmed positive results. So that kind of illustrated to them that the way to go was with the more sensitive real-time PCR method. But we didn't stop there. So what we did in the next step of this study is we took the samples that were positive by our real-time PCR method, but were negative by the melt curve method, and were non-confirming by culture. And we ran them on additional platforms. And then alongside of all the non-confirming samples, we also ran 2 that confirmed, but were also negative by the melt curve assay. And so what we found when we ran them on another real-time PCR platform, as well as an isothermal platform, we saw pretty good agreement between these new platforms and our original result. Now you'll notice there's a couple samples that didn't agree, and we'll talk about those in a little bit. But by and large, they agreed more with our result, and really the melt curve and the culture results were the outliers. And we didn't stop there. The next step we did was we sent all of these samples for whole genome sequencing. And of course, the spoiler is here in this last column, where every single sample by whole genome sequencing was found to have Listeria present, confirming then that these results by our original PCR method were the correct results. But I'd go even a little bit further into this data to point out that if you look at the two samples that did confirm by culture, and you look at what species were the most prominent within that enrichment, Listeria was one of the most prominent species, and therefore it was able to be detected by culture. But if you look at the remainder, the ones that did not detect by culture, you'll notice that Listeria was not one of the most prominent species. And in fact, if you dig into the statistics, Listeria was actually at less than 1% of the reads from those samples. So we're talking about 99% of the bacteria that were in those enrichments, or more than 99%, were not Listeria. And mostly it was Enterococcus, although you can see there were some other oddball species in there occasionally as well. But this lined up very well with what we were seeing in cultural confirmation, specifically that for these non-detected cultural samples, there was a lot of overgrowth by other organisms on the plate that was obscuring the Listeria that may or may not have been present. So the take-home message from this study was that based on what we saw from confirmation and combined with what we saw in the sequencing, the PCR, the original real-time PCR result, was the right result, and that the negative result by culture was actually an incorrect result. And so the reason we believe was due to high levels of background organisms. And so then you might ask, well, then what changed that took us from a 31% confirmation rate to a 100% confirmation rate? And our conclusion there was that because the plant was acting on these presumptive results and doing extra sanitizing or whatever was required to react to them, they knocked down the level of background organisms that were in their environmental samples, and therefore it became easier to confirm the Listeria by cultural methods. So this to me was a successful case study where we showed not only was the PCR that we were running the correct result and not the cultural result, but it also showed that increased sensitivity was benefiting the client. Now, as an industry, what should we be doing? Should we be trusting the PCR results going forward and acting on those? Or should we continue doing cultural confirmations and waiting for those results and only acting on culturally confirmed results? And I would say that, unless you're willing to do all of the whole genome sequencing and everything that we did on every single sample, you may never be able to know what the right answer is, which result is right for any given sample. And so again, I'll emphasize communicating with the lab and asking these questions is the key to try and make sense of what's going on. But at an even larger level, should the industry even continue doing cultural confirmations? And this was a session that was brought up at IAFP this year in Toronto. The question is cultural confirmation of pathogens obsolete? And this roundtable panel had experts from the diagnostic test kit industry. Eric Brown from the FDA was on this panel, as well as folks from academia. And I think there was some pretty good discussion about who should or should not be doing cultural confirmation of pathogens. But like any collection of experts, I think while the discussion was good, of course, there was not necessarily 100% agreement. And so there was no real conclusion reached as a part of the session. But I think the fact that this question is even being asked at this point does represent a bit of a paradigm shift, because I think 5 years ago, 10 years ago, people wouldn't even be considering not carrying out cultural confirmations. And then one other possibility going forward would be, what about exploring the use of molecular methods as opposed to cultural methods for confirmation of presumptive positives? And so one example that I would give here is the Rheonics platform. So Eurofins runs the Rheonics platform, which is a pattern generating platform for Listeria. And so the intended use of the platform is when you have a Listeria, you run it, and it doesn't go so far as to give you whole genome sequence data, because a lot of people, for various reasons, don't want that level of detail. But instead, it gives you what I'll call a fingerprint or a strain pattern. And then you can build a database of those patterns over time and determine if a Listeria sample or a sample that comes back positive for Listeria is the same strain that you've seen previously in your plant and it's a resident strain, or if it may just be a transient strain. And it helps you identify multiple strains that might be circulating in your plant. So that's the intended use, but we are exploring that within Eurofins as using it as a confirmation test for Listeria. And early results have been promising in that clients that have a poor track record of cultural confirmation that use this for confirmation in place of culture have seen their confirmation rates improve. But at this point, it's still, I would say, in its early days for use in that method, in that manner. And in fact, at this point, that platform is not AOAC validated as a confirmation method. So it's not something that's official, but I do think that there's a lot of potential going forward to use methods like this in place of cultural confirmation methods. And so that pretty much wraps up the points that I wanted to make today. And so I just want to summarize what I hope you take out of this presentation. And that is, first, that the use of presumptive positive term really came from industry mistrust of these new technologies. And we still use it today, and we still trust the negative results, but we always confirm the positive results. And I would argue that, over time, rapid methods have gotten much, much better. They're much more sensitive and specific today than they were 30 years ago, but they're still not perfect, and probably they will never be perfect. But in many cases, they're actually probably more sensitive and specific than the cultural methods that we're using to confirm the results. And so in the case of non-confirming presumptives, I don't think it's so easy as to just say, well, the PCR result must have been a false positive, because I think we also need to consider possibilities where the cultural result was a false negative. And we saw that with the case study that I had shown. And so as a result, I think it's important to talk to your lab. It's important to really consider what the risks and the costs are when you're trying to decide whether to trust a cultural result or a rapid test method, and that there really is no one size fits all answer to that. And then finally, as I mentioned on the last slide, I think molecular confirmation methods right now are still in their relative infancy, but I think they show a lot of promise, and in the future are likely to be a substitute for cultural confirmation, which will allow us to not only perform confirmations faster, but also have a better confirmation rate than what we see with cultural methods. And with that, I just want to take a second to acknowledge some of the folks on our national technical team. In particular, Dan actually provided a couple slides for this talk. Erica Miller is our scientist that carried out a lot of the work that was presented as part of the case study. And then Sam Wilson is actually the lab manager of our Louisville location that oversaw the case study that I shared. And then, as always, thanks to all of our clients and our collaborators, and thank all of you for joining us for this webinar and for your time and for your attention. I think, Scott, we might have some time for questions, but if not, my contact information's there. Please don't hesitate to reach out with an email, and love to continue the discussion that way.
All right, perfect. Thank you very much, Chris. We do have some questions that I was going to throw at you. So as I mentioned at the start, Chris gave this presentation previously and continues to spark great interest and debate. So I'd like to throw out just a few common questions we get in response to all this information, hopefully round out the topic a little bit further. So with that, Chris, I'm just gonna I've got 3 questions for you here. I'm just gonna throw them at you one at a time and give you a little bit of time to respond.
Okay.
So yeah, with that, so first question. Rapid methods have continued to have increased sensitivity and are more sensitive than culture. So is there such a thing as being too sensitive and detecting levels of contamination that really aren't a risk or are not meaningful?
Good question. And I think that probably depends on your viewpoint, but I think for me, I would argue that no, there's not such a thing as too sensitive, because you don't necessarily know at the end of the day whether a result that's detected by PCR but not culture is due to low levels of pathogen that were in the food to begin with, or in the product, or in the environmental sample, or if it was due to maybe it's a strain that just grows slower and so it didn't grow to the limit of detection. So maybe it was there in the food at a high enough concentration to make somebody sick, but it didn't grow fast enough to really be detected. Or maybe the food matrix is slightly antimicrobial and inhibits or slows the growth of the organism. And so at the end of the day, the PCR is still able to pick it up, but culture isn't. And so for me, I take the approach that if there's a pathogen in the sample, we probably want to be able to detect it. If it's in there, I want to know about it, basically. Because I don't necessarily know that just because something after enrichment is or is not at the limit of detection for a method is always 100% reflective of what the starting population in the product was. And so for me, I would argue there is no such thing as being too sensitive.
Okay, it's very, very fair response. Thank you very much. Okay, so question number 2. Talked a lot about software concerns at the beginning. So when you see a PCR result that you believe is called incorrectly by the software, why does it have to go to cultural confirmation? Could you just retest it by PCR and wouldn't it be faster?
Great question. And I think we get that a lot, actually, because going to cultural confirmation does cause, several days delay, and people want the result faster. And if, to your point, if we think it's an erroneous software call, then why don't we just disregard it and move on? And so I would just say that a couple things. First of all, what determines it whether the call is erroneous or not is somewhat of a subjective interpretation, right? To think that the result better than the software algorithm, because sometimes there are true positives that are going to give you a funky shape to the curve, right? And so I think that's the first part of the answer. The second part is that you always want to avoid the perception that you're testing into compliance, right? And I think obviously everybody knows the classic example of the peanut situation where they got a positive PCR test and they just kept repeating the test until they got a negative result. And I think people are still in prison because of that, because it was truly positive, and people got sick and died from that. So we don't want to give this impression that we didn't like the result, so we're just going to repeat the test and try to test into compliance. So I think because saying a curve is atypical is by its very nature a subjective interpretation, I think it's a safer decision to treat that then as a true presumptive and push it through to cultural confirmation as opposed to just repeating the test.
Makes sense. Makes sense. As you said, trust the algorithm. So,
Hmm. Hmm.
all right, last question then. So this is kind of coming from the client's perspective. I have heard from my lab that the PCR result is indeterminate. Is that the same thing as presumptive that doesn't confirm?
It's not the same thing, but in a lot of ways, it has the same net effect. So, in the case of a presumptive that doesn't confirm, right, the PCR result calls it positive, and then it goes on to culture, and the culture is negative. In the case of an indeterminate, the software algorithm basically says it cannot determine whether it's a positive or a negative. And the reason for that is usually because the internal positive control failed. And there's lots of things that can cause the internal positive control to fail. There could be uh, compounds within the food matrix or in the enrichment that inhibit the enzyme that carries out PCR. It could be that the matrix is really dark and interferes with the signal that's generated by the assay. So something like dark food colorings or cocoa or something like that might be too dark for the software to interpret the signal. And so regardless of the reason, what happens is the internal positive control doesn't work, and consequently the software says, well, the target's negative, but I don't know if it's negative because it's truly negative or if it's negative because it just didn't amplify because the assay didn't work. And so in those cases, generally we would allow for the sample to be repeated, the test to be repeated, just to see if it was a fluke. But if you get an indeterminate, on the rerun, then we have to treat that as a presumptive positive and send it on to cultural confirmation, because we can't say that there isn't pathogen in there. And so then we rely on what the result of the cultural test is to determine whether there was pathogen or not. And so a lot of people do interpret that as a non-confirming presumptive because they see that they got the PCR test and then it goes on to cultural confirmation, and then that confirmation is negative. But the distinguishing factor there is that the PCR was never presumptive in the first place. And I think indeterminates in general should be fairly rare, but they do happen sporadically. If you are seeing a lot of indeterminates in your samples, I think that's another reason to contact the lab, because that may indicate that for that sample matrix, the method's not appropriate, whether we need to change the enrichment dilutions or, maybe it's not appropriate for PCR at all. I don't know. So I think it's important to contact the lab and kind of work through, because we can make some modifications to the method so that those indeterminates don't happen. So if you're seeing indeterminates, that's definitely something to reach out to your lab about.
Okay, very good, very good. All right, so that completes our webinar then. I suppose, should you have any further questions on the topic or would like to discuss testing services, please use the link on the Eurofins webpage to contact us and get in touch with the Eurofins rep. With that, I guess, thank you again, Chris, for your expertise and presentation today. It's great talking to you.
All right. Thanks, Scott. And thanks, everyone.
Thank you.
Bye.